Okay, so it's just around 7 minutes. So it's basically a recap of all the things that we are going to be talking about in this lecture. So with this video, hopefully you can imagine lah, kind of like the things that is gonna be happening inside the plant cell during photosynthesis, both the light reaction and also the dark reaction. Okay.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
Okay, so basically this is this picture here. The reason why I post it here is because if you look at the the overall things that you have inside here with the round circle indicating the plant. So basically inside photosynthesis, the reason why a lot of people mention that plants are actually important on Earth because they are the source of our oxygen and also food. Okay, so what the plant basically needs is if you have energy, okay, the energy coming from the sun, okay, and also you must have water, okay, and you must have carbon dioxide. Okay, so actually our food, okay, glucose, let's say for like for example a vegetarian, for example, okay, the main source of your glucose, okay, which is this structure that you see here, is actually the C here is actually coming from carbon dioxide. Okay, so we basically eat, okay, we take in carbon source, okay, which is your glucose, okay, and then when we breathe out, okay, the carbon dioxide that we release is actually replenishing back the carbon that we consume in. Okay, so when we breathe out, okay, we release out carbon dioxide. That carbon dioxide is gonna be taken up by plant as a source of carbon for them to remake glucose again. Okay, which is why uh, if one day the whole planet runs out of plant, then it's gonna be a huge problem lah. Or if one day we ran out of sun, okay, if we ran out of sun, then basically all plants cannot survive because they are not able to produce their own food, not to mention producing food for animals or for human being. Okay, so let me stop sharing. Okay, so for our lecture today, we are going to talk about photosynthesis. So the video just now is basically a summary, okay, a summary of the main few points, okay, regarding photosynthesis, which normally consists of two parts. You have light reaction and you also have dark reaction. So normally photosynthesis, as the name implies, photo means you have light energy. Okay, light energy coming from where? Light energy coming from the sun. Okay, so you have this much amount of a kilojoule of light energy, okay, produced by sun that falls onto Earth every day. Okay, and 1% of this is actually absorbed by plants for photosynthesis process to actually produce your glucose. So this is the formula commonly used to represent photosynthesis process. So apart from sun, photon, you also need water and carbon dioxide. Okay, so you will know at the end of this lecture today, where does carbon dioxide enter into the pathway? Where does your water comes into play? And where is your sunlight starts to play an impact lah. But normally it's the start lah, photosynthesis, because definitely you will need the sunlight first, okay, to initiate everything. Okay, so there are basically like 10 to the power 11 tons of carbon dioxide fixed globally every year, but this figure will definitely change with more and more trees being cut down. So the number of carbon fixation tends to show a trend of reduction as more and more plants are being chopped off, which is why leading to all sorts of environmental problem or environmental issues. Okay, so of course, apart from environmental issue, if you have less plant, means you also tend to have less food, because as you can see from this formula, plant actually converts carbon dioxide with the addition of water into your glucose molecule. Okay, so if this process is being disturbed, then you can imagine the next issue will be the problem of starvation, because this is basically food, okay, glucose. Okay, and then of course, without food, without oxygen, okay, depletion of your oxygen lah. Okay, so formation of sugar from carbon dioxide and water requires energy. Okay, sunlight is the energy source during photosynthesis. Okay. Okay, so photosynthesis is a process, okay, that involves two major components, okay, which is the light reaction and the dark reaction. Uh, some call it carbon fixation, some even call it Calvin cycle. Okay, there are several names for this part. Okay, so light reaction here, basically it means it requires the presence of light. You must have light for this reaction to take place. Okay, whereas for the dark reaction, basically you don't really need much of light, but that is not a true case lah, because if you have total darkness, this part actually don't really go that well as well. Yes, so you still need light. Okay, you will know why in a moment. Okay, when we go into a dark reaction. Okay, so first part, okay, for the light reaction, basically what happens, you can basically look at light reaction over this side, okay, dark reaction over this side. Okay, light reaction, as the name implies, you require light, okay, to activate the chlorophyll that you have inside plant, okay, to release electrons, okay, activated to release electrons. Okay, at the end of the day, you end up getting the most important thing, your ATP and ADPH. Okay, with at the same time, your water. Okay, your water is going to be needed. Okay, water is basically needed to replenish electrons. So photon is going to hit onto your chlorophyll. Okay, your chlorophyll is going to be, you have photosystem 1, photosystem 2. Okay, those electrons being activated needs to be replenished. Okay, so replenishment of your electron actually comes from your water molecule. Okay, so but generally as a whole, light reaction generates oxygen. Okay, so it is from the light reaction that you have oxygen release. Okay, and at the same time you produce ATP and NADPH. Okay, so these two, so oxygen is basically a release into the atmosphere, but these two things here, your ATP and NADPH, is very much needed for the next step, which is your Calvin cycle or the dark reaction. Okay, so if you refer to textbook and so on, or other references, you will come across very often that they will say that ATP and NADPH generated from the light reaction is very much needed to feed your Calvin cycle or the dark reaction. So if this part cannot take place, basically you don't have this. Okay, and even for this part here to start, you have a protein, okay, which is by the name Rubisco, that is going to be playing an important role. Okay, we are going to look at Rubisco in a moment as well. Okay, so from your light reaction, you basically uses water to produce oxygen, and you also produce ATP and NADPH. And this ATP and NADPH is going to be used inside your dark reaction. Okay, at the same time to fix carbon dioxide, so that carbon dioxide can be utilized and incorporated for the production of your glucose, which is your sugar. Okay, so ending up generating your sugar. Okay, glucose is just one of it. Okay, after several modification, you end up you end up generating several other kind of sugar. If you accumulate, plant normally accumulate the sugar in the form of starch, which is the form that we have inside the potatoes, inside rice, and many other starchy food. Okay, so it's basically an accumulation of starch, glucose. Okay, so this formula here represents the light reaction, what happens in the light reaction. So the few components that I mentioned, okay, you need water, okay, you need NADP and ADP, and of course you need light, okay. After the completion of this, you will get your oxygen being released. You generate your ATP and NADPH, which will these two be used inside your carbon fixation step or your Calvin cycle, okay, in the presence of carbon dioxide to produce your glucose. So as a whole, this one, okay, so you have this formula. You potong sini, potong, 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 okay, it's like doing maths. Okay, then you end up getting this overall equation or overall reaction. So it's the same as the one that you saw just now, is this one. Okay, just that this time around this light is added in lah. Okay, so it's basically the same lah. If for this one you shine a light on top, you still need light. Okay, so the main player inside photosynthesis, as you have all known, is your chloroplast. Okay, chloroplast is one of the organelle inside plant tissue. It's present plenty inside leaves. Okay, and which is also why leaves looks green, because they are basically filled with this chloroplast containing chlorophyll. Okay, chlorophyll itself is green in color. Okay, so these are some of the components, okay, inside your chloroplast. Okay, you have the stroma. Okay, so in studying photosynthesis, the few component that you must remember and know how to differentiate will be stroma. Okay, know what happens inside your stroma. Okay, because there are several things. Your dark reaction basically takes place inside the stroma. Okay, so it's basically like the space around. Okay, inside your chloroplast, you have stack of this structure called granule. Okay, okay. If the whole stack of it, stacking, the whole stack is called granule. If you talk about one layer of that, one piece, this is called thylakoids. Okay, so your chlorophyll and everything, your pigment, the green colored pigment, which is light sensitive, your photoreceptor, is all basically aligned inside your thylakoid, at the thylakoid. Okay, so your light reaction actually occurs at thylakoid. Okay, so these are the description. Thylakoid membrane contains photosynthesis electron transport system. Okay, so this is where your light reaction take place. Okay, so if you recall from the previous slide, light reaction, you have the energy shining onto the 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 photosynthetic system, okay, which is basically your thylakoid membrane lah. So which will be kind of like reacting, releasing all the electrons that you want. Okay, so that is needed, and eventually at the end of the day from the light reaction generating your ATP and NADPH. So those ATP and NADPH is gonna be released into the stroma. So one common question that is being asked is where is your light reaction normally takes place and where is your dark reaction normally takes place. So just know how to differentiate which one is which. Okay, so your dark reaction normally takes place inside your stroma because ATP and NADPH are being released into the stroma, so which are needed during the dark reaction, whereas the light reaction normally take place at the thylakoids. Okay, so you have thylakoid lumen. Okay, these are basically the internal part of your thylakoid, which is aqueous interior of your thylakoid. Protons are pumped into the lumen for ATP synthesis. Okay, so proton pumping, this one we will come to this in a moment, is also one of the important requirement for photosynthesis. So stroma, okay, the cytoplasm-like on the outside. Okay, so chloroplast, you also have the DNA, RNA, and ribosome, which is basically floating very much on inside the stroma as well. Okay, the most important components of chloroplast are photoreceptors, commonly found to be your chlorophyll and sometimes carotenoids. Chlorophyll is basically the green pigment that you see inside the leaves, and sometimes you have, in some plants, you have carotenoids. Okay, different plants have different ratio of these two components. Chlorophyll is the pigment which makes your leaves looks green, whereas carotenoids is somewhat similar like your carrot, which is full of beta carotene. Okay, which is the pigment that makes your leaves looks orange or brownish in color lah. Okay, so despite that, sometimes leaves as it age, it will turn from green to orange, right? So sometimes in a country with a four season, you'll notice some trees basically they are almost like orange all the time. So still, despite that they are not green, they are able to carry out photosynthesis because they have more of this carotenoids pigment inside the leaves. Okay, so which is why they basically so basically so basically I noticed someone wants to ask a question kah? I saw anyone wants to ask question kah? I noticed pop up. Anyone? Okay, feel free to stop me anytime if you have anything to ask. Okay, so I'm fine if you want to ask anything. Okay, so and I'll just continue. Kalau tak ada. Okay, so you have chlorophyll and carotenoids. Okay, so these are just the major component lah. You still have many different kind of pigment inside plants. Okay, so this is a closer look of your chlorophyll. So this is your chlorophyll. Okay, this is your chlorophyll. Which the major component that plays an important role is actually, indeed, there is a, Okay, so there is this a question by Irdina. Okay, so does the glucose production will be affected by concentration of pigment? Strictly speaking, if you consider the based on uh, the amount of pigment, normally yes, but if you look at in terms of the length of time where a plant is being exposed to sunlight and so on, so it's basically similar, but that depends on how big or how small is the plant as well lah. So if let's say we only look at, if let's say the question that you ask is focusing on just one specific time point, then normally yes, because of course if you have more of those pigment, of course more can actually be excited, but that still depends on, okay, this is basically I would say the component that is present inside, you know, this chlorophyll. Okay, chlorophyll, you have after light shines on it, it basically becomes excited, and inside this how do you call that, photosystem, they have this photosystem 1 and photosystem 2, and in each of this photosystem, only specific kind of let me go to this one. This one, okay. So inside here, these are basically your pigment, okay, the pigment. So of course, the more pigment that you have, the more easier will the electron be transferred and eventually hit a selected member that is responsible to generate the electron that is needed, okay, for photosynthesis, which is going to be transferred along a series of electron transport, the electron transport chain system lah. So if let's say normally what it contains only one specific two lah. If you have more of this, let's say if inside here you have more, like let's say instead of this two, you have two more here, then of course you have more electron that is gonna be able to be produced. And if more electrons are being produced, more electrons will be transferred, and of course you end up generating more ATP. If you end up generating more ATP, you generate more NADPH, you end up generating more glucose at the end. Okay, so it also depends on like the size of the leaves and so on. There are several factors. You will also look at where is that? There's one more slides, but that one is towards the end. You see this one. Okay, you have photosystem 1 and photosystem 2. So it depends very much on how do they actually arrange themselves. So if you have plants that have more of photosystem 1 and 2, while you have plants that have less photosystem 1 and 2. So if the more you have, the more photosystem that is present inside the leaves, of course you are going to generate more of your protein lah. On condition if all the requirements are met. Okay, then you will have your glucose coming out lah. Okay. Okay, so now this is now going back to chlorophyll. Okay, this is your common chlorophyll structure. Okay, so this is the part normally that you look at this. This is what they call this. Where is that? Where is that word already? Profiling ring. Okay, this is the ring. So it's basically, you imagine it's like the solar panel that you have on top of your house. Okay, so it's basically like a solar panel that helps to absorb the light, the photon. Okay, you have chlorophyll A, chlorophyll B. The difference is only at this point here. Okay, if it's chlorophyll A, basically you have a methyl groups here. So if it's chlorophyll B, you have CH4 that is here. Okay, so this here is your hydrophobic tail. Okay, something like, let's say my hand here is the panel that is absorbing the light. So here it will basically be the part that is embedded inside the membrane. Okay, they are normally hydrophobic, which is the property of your membrane. Okay, so they are basically uh, being embedded inside lah, your membrane. So this is the membrane. Okay, so you have your Mg2 plus. Okay, long chain phyto group confers membrane solubility. Then you have this part which helps to absorb light. You have two major difference. So this is just like what I said, this is just the common type. You still have other different kind of photosynthetic pigment. Okay, so for example, your carotenoid, and you also have phycobilin. So like what I said just now, trees which looks yellow doesn't mean that they cannot do photosynthesis. It just means that they have this other kind of photosynthetic pigment such as carotenoid, which can also, because the process is basically to be excited and release electron. Okay, so carotenoid can also do that. Okay, so trees that looks orange in color can also perform photosynthesis with the help of other pigment rather than chlorophyll. I won't say that there's totally zero chlorophyll inside here, but the reason why it looks yellow is because you have more of carotenoid inside the leaves rather than chlorophyll. Most likely there's still some, a little bit. Okay, so the reason why plants use different kind of this or have different kind of this pigment is so that they can actually help to broaden the range of light that they can absorb. The reason why, uh, as you're aware, the reason why the green looks leaves look green because they basically reflect off a green colored light. Okay, it reflect green light and then it absorb the other. Okay, so you have other kind of pigment besides carotenoid. You also have phycobilin. Okay, phycobilin is another form, but this kind phycobilin, this pigment is not found most of the time in plant, but this pigment is found most of the time inside algae. You know your algae can also perform photosynthesis, right? So algae can also use sunlight, but not with the help of chlorophyll. They require the help of phycobilin, okay, to be the photosynthetic pigment that actually helps them in photosynthesis process. So these you can normally find this one in red algae and also cyanobacteria, which can also perform photosynthesis. Okay, so the main role or the main purpose of this pigment is basically to absorb, okay, to absorb sunlight. So like what I said just now, chlorophyll, they are basically green. Okay, they are green because they reflect off green colored light. So when the light are being reflected into our eyes, we interpret that color to be green, while those light that are not reflected are basically being absorbed, disrupt. It's being absorbed by the tree or the pigment. Okay, so if you look at this absorbance spectrum, you can see that you have chlorophyll A and chlorophyll B. So the peak here actually means that this pigment actually absorb huge amount of orange and almost yellow colored light. Okay, they reflect green color, but they absorb a lot of this orange and green range of light. Whereas these, which are also other kind of pigment, okay, which are also found inside your cyanobacteria and also algae, different from chlorophyll, this phycocyanin, for example, absorb green colored light, okay, and also slightly yellow, okay. And if I remember correctly, this pigment here itself is slightly, if I remember correctly, lah, is blue or greenish in color, blue. Okay, and then it absorb green colored light. Okay, whereas your carotenoids is orange, yellowish in color. So because it reflects off that yellow color, but the compound itself actually absorb blue colored light. Okay, so you imagine if a tree contain both chlorophyll and carotenoids, you the plant cell can actually utilize both the blue light and the red or yellow photon for photosynthesis. Okay, so which is why the more variety of the pigment that the plant has, the larger the range of photon that the plant can use for photosynthesis process. Okay, I have a question here. Okay, so does this mean carotenoid increase as the tree grows? It's more not to say that it increase because um the percentage to me the percentage is almost fixed unless if I'm not mistaken, because as uh, tree grows, for sure it grows, okay, it also age, because as it age, for sure once it depletes or degrade, then the color fade. So it could be because that the amount of carotenoids increase, but I doubt if that's the case. Okay, so because the amount of carotenoid or uh, chlorophyll that is present is already kind of like controlled by the gene that is being expressed by the plant itself. Okay, so it's like already being encoded into the genome sequence of the plant. So I I believe that uh, it is fixed lah. It's more of a being fixed rather than changes from time to time. But however, there is a possibility that this part I'm not sure. This point, the point that I'm going to mention is I'm not so sure, is especially plant in fall season. You notice that you have certain trees that looks yellowish all day round, right? But sometime of the year it will kind of like change color. Initially it is green, sometimes it is orange. For those kind of plant, yes, most likely they have inside the genome system, in its gene, most likely there is this regulation system that control the amount of carotenoids that is expressed at certain time of the year, and the amount of chlorophyll that is expressed and produced at different times of the year. But this one, uh, to be honest, I'm not so sure lah, whether if there's actual change, but I believe there might be. Okay, the plant has a wide option to absorb other wavelengths of light if it has both pigment, right? Yes, correct. Okay, so Edina, the plant has a wide option to absorb other wavelength of light if it has both pigment. Yes, correct. So the more, let's say if you have a plant that is capable of absorbing carotenoids, no, a plant that contains carotenoids, chlorophyll, but at the same time, they also have phycocyanin and phycoerythrin. And yes, of course, you basically look at the hump. Okay, the highest peak indicates the range of wavelength that the pigment absorb the most, the photon. Okay, if before this, most plant normally have this chlorophyll and also carotenoids, right? But these two components are normally found only inside algae and also your cyanobacteria. So if one day your tree can also have this pigment inside the tree, then yes, the tree will also be able to absorb this range of light. Okay, so which is the more the better lah. And actually, if you go in deeper, right, there are actually research that looks into the effect of light that is being shined on top of tree leaves. Different time of the day will actually have a slightly difference in term of the amount of red light or blue light. But this one, I just kind of like come across several years back lah, when I was looking up for some information. So there are actually a different amount of blue light or red light uh, available throughout the day. Okay, so okay, feel free to ask any questions if you have more to ask. Okay, so harvesting. Okay, now before this, photosynthesis. Okay, so photosynthesis, of course, the plant use light. Okay, the plant use light, so it absorbed by what? Absorbed by the chlorophyll inside the plant. So once it is absorbed, basically what happens is you have a lot of this chlorophyll that is being arranged. Let's say we talk only on chlorophyll lah for now. You have chlorophyll, you have antenna, so basically it excites, okay, the chlorophyll. So the chlorophyll basically helps to transfer the electrons to this special pair. Okay, photosystem contains special pair chlorophyll molecules that undergo charge separation. So charge separation and donate electron to the photosynthetic electron transport system. So eventually they have to transfer the electron, excite, okay, to a pair which is called, this is basically inside photosystem. Inside a photosystem, you have photosystem 1, you have photosystem 2. Okay, so what happens inside that photosystem? When lights hit, the energy is going to be directed, okay, to this special pair for charge separation. Okay, so what is charge separation? Yeah, if you are interested, you can actually look at this 2 video as well. Okay. Okay, so charge separation is basically the production of a molecule which is the donor, which is positive, and the acceptor, which is negative. Okay, so what it means here is basically, if you follow this sentence here, okay, donate electron to the photosynthetic electron transport chain. Okay, so donate electron. So the electron comes from the special pair. So once the special pair receive the energy that they want, the electron there is gonna be excited. So you see, normally under ground state, you have electron pair 2. Okay, so which is indicated by the 2 line that you see here. Once it gets excited, it will go up, okay, into an higher energy level, so which is called an excited state. Okay, so once it got excited, it cannot be excited forever, so it's gonna pass that excitement, okay, kind of like excitement, to the next chlorophyll. Okay, so which is of a lower energy. So normally the passing of energy can only be uh, occurring between molecules which are of similar energy level or of a lower energy level. It cannot be from this stage to a higher energy unless you supply energy to that process. Okay, if not, normally it is going to be transferred to an lower energy. Okay, so it's going to be transferred to an acceptor lah. Okay, so with the by receiving an additional electron, so it becomes negative. So this one lose one electron, so it becomes positive. So basically this thing happens a lot inside the photosystem. So one got excited, it starts to kind of like a release of electron, so forming this charge separation. You have one positive, one negative. Okay, so energy transfer through antenna pigment system by resonance transfer. So this is what it meant by like the one that I mentioned, resonance transfer. It can only be transferred to one that is of similar energy level. You look at the horizontal line here, similar energy level, or it can only be transferred to those of a lower energy level. It cannot be transferred to anyone that is of higher energy level. No, cannot. Okay, so in order for energy to be transferred, those molecule needs to be arranged as close as possible. If they are very much far apart, then the rate of energy transfer basically decreases. So this point is very easy to understand, whereby you imagine, would it be easier to pass a ball to someone who is sitting beside you as compared to passing a ball to someone who is very, very far away from you? Okay, you have to toss that ball very far, right? So you have to gather that much amount of energy in order for you to toss that ball that far. So of course, the closer you are, the easier for the energy transfer to take place. Okay, so this process is very much similar to your electron transport chain system, which you have studied last week. Okay, so in the process, you basically generate ATP with the help of ATPase. Okay, some people call it ATPase, some call it ATP synthase. It's the same thing. Okay. Okay, so generate proton gradient, so which is very similar to your electron transport system that you have studied last week. Okay, so this is the one for plant. Okay, now as you can see here, this is basically photosystem 2. This is your photosystem 1. Okay, photosystem 2, you have P680 and then P700. Okay, the number here actually indicates the wavelength that this photosystem likes the most. Okay, if you refer back to this one, okay, so phycoerythrin, it likes or it absorbs the highest at around maybe like 590, around 590 like that. Okay, whereas carotenoid, if you see the hump, okay, you have two hump means it liked these two wavelength. Okay, so I would guess this one is about 430. This one is about 400 and maybe 480, for example. Okay, so for photosystem 2, the chlorophyll inside here, it basically likes the wavelength of 680 nanometer, okay, photon. Okay, whereas photosystem 1, it likes 700 and 700 nanometer. Okay, so what happens here? You have photosystem 2 and photosystem 1. So photosystem 2 here is basically normally if you refer to any reference, you will see the process starts from photosystem 2. Okay, photosystem 2, and photosystem 2 is where your water molecule is going to come in. Okay, so you know so far that here, once you have light shining onto it, you have electrons that is being gonna be excited and released by the chlorophyll molecule. Okay, so that electron is gonna be transferred. Okay, which is why in the earlier slide it is mentioning that it is very much similar with your electron transport chain system inside the mitochondria. Okay, so it's basically going to be transferred, transferred, transferred. At the end of the day, that electron is going to end up to your NADP. Okay, still remember inside your light reaction you have two kind of like two things produced. Okay, one will be your ATP and another one will be your NADPH. So that electron is gonna pop, 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 go and end up being taken up by your NADP. Okay, so NADP is going to be reduced into NADPH. Okay, so you end up getting NADPH. But this part cannot forever be keep on producing electron, right? So someone need to replenish that electron for the missing electron that is being excited and released out. So who is going to be replenishing that? Your water is going to be the one replenishing. Okay, your water is going to be the one, so that is going to replenish the missing electron. You see, water donate the electron here so that the electrons can be transferred along the way. Okay, so which is why uh, this process is also called photolysis. Okay, so your water molecule is being chopped up. Okay, so with the help of your light. Okay, so electron is going to be donated here, pass along the chain. Okay, so as it pass along, before it reach photosystem 1, you will come across this structure called cytochrome Bf complex. Okay, so it's basically two components. You have cytochrome B and cytochrome F actually. Okay, you have in between here, you have your plastoquinone, and then you have plastocyanine, which is all going to be inside. Plastocyanine here, okay, is here. So plastoquinone is going to come, transfer the electron and everything. So as it transfer, passing through here, this process is going to lead to accumulation of hydrogen. Okay, so this one is the stroma, the outside part of your chloroplast. Okay, I know. This is thylakoid, the thylakoid membrane. Okay, thylakoid membrane. So this is the stroma. So if you imagine this yellow, this one will be the membrane of your chloroplast. Okay, so this is the thylakoid membrane. Thylakoid membrane, and this side, this is the lumen. Okay, which is why I told you just now. Okay, it is important that you know at which part the reaction take place. Okay, stroma is the one outside, lumen is the one inside. Oh, so it's not written here. Okay, the 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 dark line that you see here, you have basically like a sandwich like this, right? Lumen is the one inside, lumen. Okay, so in this, okay, so this is the lumen. You have to imagine, okay, so eventually as light shines on this, okay, more and more electrons is going to be passed on, okay, with at the same time with more water molecule being chopped up with the release of your oxygen. So oxygen is going to be released, electron is going to be passed on. So as it is being passed on, by the time when it reaches cytochrome BF complex, this here is going to cause more of the process of transferring through here is going to cause your hydrogen to be accumulated inside your lumen. Okay, accumulation of lumen, accumulation of hydrogen inside the lumen is important for your photosynthesis process. That is one of the requirements. Okay, as you have more and more H plus accumulate inside here, as the condition inside here basically becomes more acidic. You have more acid, you have more H. So this part becomes more acidic, while this part becomes less acidic. Okay, so this is one of the requirements, okay, that is needed for the next stage. We will come to this point. You just remember there will be an accumulation of acid inside the lumen, while the outer part becomes less acidic. Okay, so this is one of the requirements. Secondly, there will be this process where you see magnesium is being pushed out from the lumen into the stroma while chloride comes in. Okay, accumulation of Mg2 plus is important, especially inside your dark reaction. Okay, we will come to that in a moment. Okay, so this is one of the second requirement for your photosynthesis reaction. So as it pass along, eventually, you know, this is your photosystem as well. You have a pair of chlorophyll, which is capable of releasing the electron. So once the electrons are released, here there is no water molecule to be chopped for to replenish. So basically the electron needs to be replenished from starting part here. Okay, so from here it is basically going to be transferred along here. So the one that is excited is going to go out, and this one is going to come in and replenish, okay, the missing electron. Okay, with time, as more and more this H plus is being accumulated here, the gradient that is produced, okay, because eventually as more and more H plus accumulates here, this one becomes more acidic. Okay, you have a higher concentration of H here, less concentration of H inside the stroma. So this condition will actually allows your ATP synthase to, the difference in gradient is going to power your ATP synthase to produce ATP. Okay, so this is basically what I mentioned lah. Okay, photolysis, so basically the things that I mentioned. Okay, you can refer to this. Okay, this one I'll come to this in a moment. Okay, where is that? Okay, before I go to this, where is that? This one. Okay, so you have your accumulation of with time. Okay, as electron, this is a light-driven ATP synthesis because you need light. You need light to power the electron transport to actually causes the accumulation of hydrogen inside there. So electron transfer through the proteins of the G scheme. So this one is the one on top, generates a proton gradient across the thylakoid membrane. So this is gonna power, because eventually, of course, this needs to go out. Okay, of course, you cannot keep on increasing this without releasing the stress. Okay, so to release, so H is gonna pass through this ATP synthase. So passing of this ATP synthase is gonna activate the phosphorylation of your ADP into ATP. Okay, so which gives rise to your ATP being produced. Okay, So that is one of the requirements. So this is the process which is called kind of like it is being represented in a scheme called Z scheme lah. Z scheme. So if you look at this, so basically this part and this part is basically representing your photosystem. Okay, so from photosystem 2, okay, to photosystem 1. So as you have light shining onto your chlorophyll, electrons are being excited, and electron will be, once it is excited, of course, it is being excited to a higher energy level. So eventually it is going to be passed on to the next molecule. So each of these will have a different reduction potential, okay, as indicated by this here. So the higher they are, the higher the tendency they will, or this molecule will be able to donate the electron to the next. Okay, so which is why uh, the lower they are, the easier they tend to accept uh, electron. Okay, as you can see based on the plus sign here. So say not, say not, say not electron, say not electron, say not electron, something like that. Okay, so they are basically being passed along a chain of reaction, okay, pass on from one molecule to another, and eventually back to photosystem 1. Okay, so here you have cytochrome Bf, which causes the accumulation of 8 hydrogen. Okay, so there will be this. Okay, so this is basically like starting back from the front. So you have light energy activates this. Okay, so you release off electron, and this whole process actually requires 8 photon. 8 photon. So this is talking about 680, the first photosystem. You are aware that you need water molecule, as this one keeps on releasing electron. You need to replenish. So one water molecule can only donate 2 electron. So in order for you to generate one oxygen molecule, you see 2O here, you need 2 water molecule. Okay, so which in the process you will need to release 4 electron. Okay, so only then you end up generating one oxygen molecule. Okay, so meaning to say you need to have 4 photons hitting onto that part, then only then you have one oxygen molecule. The energy release is in the form of electron. Yes, Nisa. Okay, so a question by Nisa. So the energy release is in the form of electron. Yes, so it's basically more like not to say in the form of an electron. It's more like because you transfer electron from one to another, right? When you transfer from A, you transfer to B. In the process, you will have energy that is being released, which is why electron transfer can only occur if it's of the same energy level. You see, this is energy level, okay? So it is Nisa, your sentence is more like terbalik. Okay, so electron transfer involve the release of energy. Okay, so which is why electron can only transfer to kawan sebelah, which is of similar energy level, or to kawan sebelah dekat of a lower energy. Okay, so of a kind of like in a lower energy state lah. Okay, Nisa, saya baru perasan Nisa punya soalan. Okay, that's 255. Okay, so now going back. Okay, so it is being transferred along until here. It is being excited back again because here you have light energy. It goes back up, and then eventually it is going to pass along a series of chain until end up being received by an ADP. One of the common question that is asked will be, siapakah penerima terakhir elektron dalam photosynthesis? So just take note, your NADP is your last receiver of your electron that is being excited and released inside the electron transport chain. Okay, so next. Okay, so this one is just now. Okay, so this is one of the member inside your electron transport chain, and you have purified team A and plastoquinone. So these two member are the two member that is located here. Okay, are the two members that is located here. Okay, so basically it talks about the property of this. So one plastoquinone here can actually hold a maximum of 2 electron and 2 hydrogen, which is why sometimes you will see in books plastoquinone is labeled plastoquinol. Sometimes they call the one with H, the one with H2H, they label this as PQH2, called plastoquinol, NOL. Okay, plastoquinol. So they are able to carry, because what you see here, these are actually carrier. Okay, they help to carry the electron from here to here. Okay, so these are carriers. Okay, so from your, you see, PQH2, so it can carries and then pass through here, and the process actually leads to the accumulation of 8 proton. Okay, if you go through this diagram, you will see that first you have your PQH, okay, PQH2, okay, if you look closer, there's a small 2 here. PQH2, your plastoquinol here, which carries the 2 hydrogen and 2 electron. Basically, this plastoquinol here will not donate, it has 2 electron, it won't donate both of the electron to ferredoxin no, to cytochrome F, cytochrome F. So cytochrome BF complex here is basically consisting of 2 components. So it's basically consisting of 2 parts. You have cytochrome B and cytochrome F. Okay, so the electron is basically pass one of it to cytochrome B, another one to cytochrome F. So cytochrome F with this one electron is going to pass to plastocyanin, and plastocyanin is going to carry this and pass it to your photosystem 1. Okay, so here uh, 2 of the hydrogen is being, you have PQH2, right? So this H2 here is going to be released. The moment the 2 electron is released, the 2H2 is basically going to be released, and eventually this one will go back, and it's about dah tak ada H2 kan, so it becomes like this. Okay, so there will be another H PQH2 that comes along, okay, and that is gonna donate 2 more. Okay, and one of it is gonna be eventually passed back to this, the one yang tengah dekat tunggu dekat tepi ni. Okay, one of it is eventually gonna be passed back to this one, and with time, this is gonna collect more and more, and eventually it can form back this. Okay, by collecting more of this hydrogen from the stroma. Okay, so once it collect the H, 2 more H from stroma, it basically is because basically this thing is jalan-jalan lah dekat dalam ni. So eventually once it forms PQH2, it's gonna go back and starts the cycle again, and eventually with time, as this process repeats, uh, the H from the stroma is somewhat like indirectly being transferred and accumulated inside the lumen. Okay, so which is why as a whole, if you have because basically from the process here, you will have 2, okay, 2 PQH2 is produced from 1 oxygen. So from this 2, you will actually causes the accumulation of 8 proton that is being accumulated inside here. Okay, so which is why if you look at a lot of these figures, right, like for example this one, it always indicates 8H plus. Okay, if you look at the previous one, you also have 8H plus. Okay, so it is basically going to be passed along, and causes, as it passes along, it causes the accumulation of this H plus. So this one is towards the end. So eventually after passing photosystem 1, photosystem 1 is gonna, see, photosystem 1 is gonna pass to ferrocene. Ferrocene is gonna then pass it to NADP to produce your NADPH. Okay, so this is basically this one. See, so NADP is the terminal acceptor, electron acceptor. So it is the last person or the last molecule that is gonna be receiving the electron. Okay, so this is your photophosphorylation, which is the production of your ATP by phosphorylation of your ADP into ATP. Okay, so this is the structure of your ATP synthase. Okay, so this is basically some of the basic structure. Okay, so ATP synthase, chloroplast protomotif. Okay, so in the process, you end up generating 3 ATP. Okay, so 3 ATP is going to be generated per molecule of oxygen. Okay, you will have 3 ATP from one oxygen molecule. Okay, and in the process, you will generate 2 NADPH. Okay, so 3 ATP, 2 NADPH per molecule of oxygen. Okay, and this last one here, okay, so basically it tells that you have photosystem. Okay, now it is best to explain using this one. Okay, so in most textbook or most figure, if you go and Google, you will find a lot of pictures like this. Okay, where you see photosystem 2 on the left, followed by photosystem 1. In actual fact, this thing here is, you know, your membrane is fluid, okay, with fluidity. So basically, this thing can move here and there, here and there. So it's like very fluid. It's like a boat on sea, right? You have a sea. Okay, boats basically they are free to move here and there depending on where they want to drive or where they want to head for. So same goes for this one, whereby they are basically uh, I would normally imagine this to be floating. So this photosystem is very much like floating on a fluid sea. Okay, uh, which is why if you look at this one, okay, if you look at this picture here, so this is your thylakoid. So this is your lumen, this is your lumen, and this is your stroma. So if you follow the color indicator here, you have photosystem 2, photosystem 1. Okay, so depending on the light condition, okay, light harvesting antenna, okay, abbreviated as LHC. So this antenna here basically is your chlorophyll lah. Okay, are also present in the lamella, okay, which is this one, okay. Okay, and then under low light condition, when the light is low, basically your this one, your photosystem 2 is gonna be very much associated with the your antenna, because when the light is low, you have low amount of photon. Okay, when you have low amount of photon, you know your LHC here is the one that is receiving the energy from light, converting light energy into chemical energy. So this, of course, it needs to transfer to photosystem 2 first. So of course, under low light energy, your LHC is basically gonna go near to your photosystem 2, so that it facilitates the energy, the electron transfer process. Okay, so but if kalau let's say light energy a lot, so normally they are very much further apart lah. So basically, this picture here tells you that although in textbook you see photosystem 2 on front, photosystem 1 at the back, so this thing is basically very much randomly aligned, because it's basically a 3D surface. You have to imagine it's 3D surface. It's not electron moving from left to right only. It could be electron moving from front to the back, to the back to the front, to the left to the right, and so on. So as long as the sequence is correct. Okay, you have photosystem 2 moving to photosystem 1. So it could be photosystem 1 on the back rather than photosystem at the side. Okay, so basically you have to imagine lah this part. So they are very much fluid. Okay, it can be at anywhere. Okay, so depending on the condition lah. So if light is low, they are very much closely aligned. If light is high, so they are much further apart. Okay, so now we are at 3:18. So how about 15 minutes break or 10 minutes break? We come back here at 3:30. Okay, to prevent any one of you